Hello, everybody. My name is David Leclerc. I am a host of this new podcast we got started called Tools of the Trade, where we basically try to learn more about our partners in industry, new companies, new groups that helps us with our research. I am here with Daniel Vossel, uh, who's going to be my co-host and ask really, really smart questions. Hi, Dan.
Hey,
And I'm super excited to have the people from Ozette with us today. Greg Finak is CEO co-founder. Hi, Greg.
hi, David. Hi, Daniel. It's really great to meet both of you. Really excited to be here on the podcast with you guys.
Pleasures are on our side. And we also have Corrie Ortega, who is Chief Product Officer.
Yeah, really happy to be here with both of you and talk more about cytometry data.
Well, thanks so much for making time to meet with us. Look, the full idea of the podcast is basically we want to meet with these groups that we hear about, but for one reason or another, we just don't get the deepest contact with. So we're always kind of aware of your presence, but not exactly sure how to interact or how to basically make your group benefit the research effort in our facility. So the podcast is basically there to like bridge these link between our worlds. And so thanks for coming, Greg. I'm gonna ask you to get started by introducing Ozette. I've noticed in your website that you've done your PhD in McGill.
Yeah.
So as a French Canadian myself, I know that you had a blessed life up to that point. But like, can you tell me a bit of what happened after and how Ozette can come to be in this world?
Yeah, glad to do it. So yeah, I did spend quite a lot of time in Montreal. Went to McGill, did a PhD there, did a postdoc with a really fantastic guy named Raphael Gottardo, who used to be at the Fred Hutch and had the opportunity to come to the States when he moved there. And that's where I started working on flow cytometry data before, people were calling it single cell data, right? It was just flow cytometry data. And it was, you know, like like most like most things, you look at a problem of like trying to solve this problem of like doing computational flow cytometry and you think, well, this should be easy, right? It's just clustering or whatever the case may be. And I think everybody who's probably approached the problem for the first time probably had the same thought for like the last 30, 40 years, whatever it is. It's so much, obviously, so much more complex. The system is incredibly complex. And so I spent, most of my career at the Fred Hutch working on this problem, building tools, open source tools, for R and bioconductor that people could use to help analyze their data. And there was always this problem of, you needed to be exceptionally like skilled in writing code and doing things like that to do anything with these tools. And you overlay on that the fundamental like biological problem is that the data is getting increasingly higher dimensional, increasingly complex, and we're trying to pull out things that are exceedingly rare, and hopefully they represent real biology, and there's some biomarkers there. And then the challenge was always like, well, if we can do it once, well, will it reproduce, right? Are we pulling out real signals, right? This is kind of the meta problem that you face, whether you're doing flow or whether you're doing genomics or whatever the case may be. And with my co-founder, Evan Green, and Raphael, at one point, we kind of like just stepped back and said, look, there has to be a different way to think about this. And Evan put together an approach that kind of moved away from sort of the traditional sort of machine learning, clustering ways of thinking about finding signals in flow data. It was entirely non-parametric, incredibly high computational cost, but to our surprise, I guess, because these things don't happen a lot, it worked incredibly well. And we were starting to find like biology that we could then reproduce in like similar studies, pull out the same signals, find the same, like, find the same kind of biology, the same kind of correlations that, would predict things. And this kind of happened over and over again on a few different projects we were working on. And we thought, well, this is not going to be something we're going to be able to put out as just an open source tool. It's far too like complex to work. This needs to go farther. And that was sort of the core idea of founding Ozette. It's to make incredibly powerful tools available to the community, but to make them accessible not just to computational scientists, but to biologists, wet lab people, so that they can answer questions that they want to answer with the same level of like certainty that they might get if they farm it out to their bioinformatics team, who might spend a month, 2 months on it, and then like come back, and then you spend your time in front of the email going like, well, what if you look at it this way, or what if you do a figure like that, and how do I get back now from these like incredibly high level views of how immune populations behave across my cohort to like dive into this particular sample to make sure that the data is actually gated in a way that makes sense. And so that's really what Ozette is all about, kind of taking the sum of tools and knowledge around computational flow and wrapping it in a package that's performant and that's accessible to everybody.
Awesome. Corrie, do you want to jump in and let us know how you got involved with Ozette and what's your role and function over there?
Yeah, absolutely. I joined the team early. I am one of the day oneers, and it's been an amazing adventure since I joined Ozette. And one of the things that got me really excited, I think, is exactly the way Greg described it. There are these really deep pain points that are really tension between instrument advances and not having advances on the analysis side that are really matched with that progression. And we're seeing higher dimensional panels, higher dimensional data. We know the power of those data for decision making in real life with clinical trials with patients on hand. And I think that there's something there that we want to do right by that ultimately, by all the way from discovery, all the way into clinical applications, and really harness the power of the data that we're all investing in. And there was opportunity for us on the analysis side, as Greg was saying, to build on the foundation that he and Evan and Raphael built at the Hutch and really turn it into accessible packages, all the way from unmixing or raw data coming out of a spectral instrument to the full annotation and analysis of those data, either through targeted analyses or true discovery. And that's, from canonical to rare to sometimes uncharacterized, right? It's really being able to explore the data in the deepest sense, but also in the most simplistic, which I feel like scientists deserve. Oftentimes we have to use software where the UI is not great or intuitive, or it's limited by your expertise with R or, coding background. And here we're trying to make it available to all cytometrists or teams that associate with them, so that they can all look at the same data and see sort of the tangible value or the value in those data, right, for decision making.
But what do you do?
Oh, yes. Great question. I wear many hats at Ozette, and I started on the operations side, but always worked really closely across all teams. And now I help take care of our product team. So I help package and inform a lot of the products and services that we provide to different scientists and different teams, all the way from academia to pharma to contract research organizations. And so it's hearing a lot about those pain points from our internal team, from customers, and understanding where we can make investment and add value and enhance those technologies really for the best customer experience, but also in really getting the most out of not only each individual data set, but across data sets as well for any scientific experiment project team.
I mean, what is Ozette doing for, like, average shared resource lab? If I'm a one person core, small university, Why am I thinking Ozette?
So a lot of our customers that do volume, like a shared resource lab, might have certain panels that they kind of have on the menu for their customers, right? And if you think about what is the gold standard for, let's just talk classic bread and butter, I have to gate my data, with some kind of tree. The gold standard remains hierarchical manual gating. Why? Because doing it, well, because if you're, I think there's a few reasons, right? In the clinical space, it makes sense because if you're doing something computational, it has to be reproducible and you have to be able to validate it. And that hasn't yet, so far been possible with the existing computational tooling that's out there. This is a problem we've solved. Leaving that aside, in your typical academic space, I think most folks want to see it, right? And clustering is like a little bit of a black box. And so it kind of comes back to the tooling that's provided and the UI and like, can I program? Why do I have to program? Nobody wants to do that. They just want to like have some guarantees that the way they want to gate their data is the way their data is going to be gated. And the only way to do that, so far until now has just been good old classical hierarchical manual gating. And so I don't know if your lab gates data for your customers or if your customers come in and do it themselves. But one of the ways that we help with this particular problem is we have, packaged a tool that we call Ozette Endpoints that automates the process of hierarchical gating. It's scalable. All you have to do is describe how you want to actually gate all of your endpoints, right? And how do we describe endpoints? Well, typically one line, right, is going to be an endpoint, and you write down your markers and you say, I want to gate, lymphocytes in forward scatter and side scatter to get singlets, and then I want to gate my lymphocytes, and then I want to gate CD3 and CD4, and then I want to gate CD8 or whatever, right? And you build this thing. People already do that. And then they have to go in and they have to draw boxes. Well, the way we sort of help folks with that is like, well, okay, just pre-specify what it is you want to do. You feed that into the platform, and the platform is able to then turn that into a tree, a gating tree, and it learns from the data that you feed it where, using some of this technology that I talked about that Evan invented when he was at the Fred Hutch, to then be able to correctly actually computationally draw those gates throughout that entire data set across all of your samples, and then leveraging the platform to visualize all of that, really quickly, review your data, make sure you don't hit any sort of QC issues, identify those QC problems before it's too late, which is very often the case, right? One of the sort of fundamental problems is that you'll have data quality issues, but you're not looking at the data. Like you can't look at all of the data. And so you kind of get to the end, and then the scientist whose data it is like, wait, there's something wrong here. And then you have to go back. And Ozette provides a framework to really give you the ability to see the problems with the data at all of the steps before you sort of get to the end. The gating is automated. And then you have, high reliability that, the endpoints that you pull out are going to be the things that you're actually trying to measure, and you're not accidentally, dealing with spillover spreading and markers sort of being artificially like positive because you're actually just looking at spreading and whatnot. So various things like that. But it's a sequence, right? It's a sequence of steps that we've tried to kind of abstract away and ensure that as you go through this, process, you're getting your eyes on the right data and looking for, the problems that might be there at every step before they kind of get out of hand, so that you have high confidence that you have high quality data on which computational approaches can then work correctly.
So I gotta put the data in. I gotta do a little bit of training, where I go, yep, that gate's right. This gate needs tweaking. This is not right. Then I apply it on a larger data set, and it's got some metrics in there that go, this file is absolute junk. Don't use that file. And this, is potentially good and bad. And then we get a good visualization at the end on these are what your overall populations are. But also relays it back to hierarchical gating too, if I want to double check things.
Yeah, that's in a sort of high level view. That's a good way to think about it. It's a mix of different approaches. In some places, we're doing training. For example, your cleanup gates. We found people want to do very specific things depending on what kind of samples they're working with, and so that's an area where it allows for a great deal of customization. You can gate a few samples. The platform is smart enough to be able to look at your entire data set and pull representative samples out of there that you can use for training, so that you don't have to gate everything, or you don't have to take a best guess at sampling. It'll actually sample smartly for you. You can gate 5 samples by hand, and then it'll train on those, it'll learn from those, and then it'll apply it to everything else. And then you can quickly review, what it's done, make some modifications if you need by hand. And, we kind of audit log everything along the way. So if there is manual intervention, you're going to see that in your report. You're going to be able to know this gate or this sample was a little bit different, so we had to tweak it. All these kinds of things, which are really valuable, certainly if you're working in the clinical space. But primarily, for a core lab, once, if you're kind of running the same panel and a pretty similar gating tree all the time, once you've trained it, you've set it up, right? You can just run and go as your samples come in from your customers. So you save a lot of time, right? In sort of the long haul, in the follow-up experiments and the follow-up batches of samples that come in.
Is there really a threshold then where that's useful? Like that you and David, that really, you should be doing 20 color panel or more, that, if you're just doing 4 colors on a routine basis, this, isn't for you. Or there those kind of markers, or this is probably more appropriate for you?
I mean, I think that's probably true to some extent. If you're not doing a large volume and you're not doing high dimensional data, probably the tools that you have today are just fine, right? I don't know that you're going to see massive benefits if you're running 4 or 5 color panels that, are very simple to gate. But that said, we have looked at experiments with 6 to 8 color panels, for T cell populations, where we found signals and biomarkers of response and crazy polyfunctional T cell populations that predict things that the original sort of designers of the panel and the folks who ran those studies initially didn't think to look at. There are sometimes interesting combinations of markers, even in relatively low dimensional space on cell populations where you wouldn't think to look, right? We're running these really high dimensional panels on the immune system, which is unbelievably complicated. As much as we know about it, we don't know nearly as much as we think. And there's a lot of really interesting signal in there that we just haven't had the ability or the tooling to look for.
Yeah, I would say in partners that we've worked with to date, we've seen a range in panel size or dimensionality. Oftentimes we see things in the 10 to 15 color, and I think there's still a lot of utility for Ozette solutions in that range, to the examples that Greg gave. And then we work all the way up into the 30 plus, 40 plus. Ozette has a 40 color panel. And so I think that's where, we often see it's an increasing value add, I think, as the dimensionality increases, obviously. But more than once, we have worked with partners who have observed missed signal from previous analyses, and sometimes in very surprising, I think, outcomes.
Can I ask a question about training a model? And I'm neophyte, in terms of AI, but how much time would it need to train a model to whatever you want to do? And if you get different people trying to train the model on this similar thing, how divergent the data or convergent or how similar the data would be down the line?
Yeah, great question. So there's a few places where we rely on model training. in the platform. One of them is in, like I said, when there's just very sort of bespoke and custom, like we call it like pre-gating or pre-processing of the data, where you're trying to like get rid of, clean up your events that come off the instruments to get to like those pure, what we call parent populations, which you're eventually going to start looking at to identify, let's just say T cells, right? You want to get to things that are or CD3 positive and whatever else. And people will insist on, well, this is the way I have to gate to get to those T cells. So, we're not going to come in and say, well, you should do it this way. Like they insist on doing it, their very specific way. And so fine. So we provide them the ability to do that. And then we train models on gates. And the way those work is, they look at the distribution of the data. And in order to pick the right model, we might have a lot. We're going to find a model that has been trained on a sample that looks a lot like the one we're about to predict. So we kind of like look against our library of models and we're like, okay, this one has a very similar distribution. And, it was used to gate, singlets or whatever. I don't know, maybe not singlets, but it was used to gate whatever it was used to gate, that pair of markers. And so we're going to pick that model because we think it's going to do a really good job. How long does it take to train? Under an hour, you can do an entire, fairly complicated tree. And that's for the pre-gating. Endpoints itself, very different beast. It's not pre-trained. It really is sort of data-driven, I would say, like pseudo-Bayesian framework where we rely on prior information about what the entire set of samples look like and the distributions of the markers and, where we think positivity is for each of the markers, whether it's like bright or dim or slightly positive. And we kind of like partition the space of those markers across the entire data set and then rely on that to infer a best guess on incoming samples where those boundaries between positivity and negativity should be. Once we know that, then we're once again like relying on something that looks a little bit like clustering within a much more constrained space to like draw those gates, so that they respect those boundaries. And then, of course, you get the chance to look at it, right? And so in the end, again, you're looking at a tree. You can kind of click around, look at your dot plots. These look right. This one maybe doesn't quite look right. You'll have your distribution of all the sample statistics for the different populations. You can quickly look at outliers and just kind of get to the bottom of it very, very rapidly.
I have so many more questions.
Go for it.
No, please, before I take us on some wild tangents.
I was going to switch to Unmixing. So your last chance, Dan.
No, I want to hear that too. So
okay, cool. So Ozette I've known because I've watched a couple of webinar dealing with Unmixing. And so Unmixing is kind of we still talk about, do you apply computation on top of your Unmixing? I use that a lot on my other podcast because I think it's funny at this point because it just won't go away. What's your view on Unmixing? What does Ozette brings to the table?
Yeah.
How do you see the problem?
Yeah. The way we landed on our approach to Unmixing, we kind of backed our way in, right? We started with, we have a solution for discovery and everybody's like, oh yes, discovery, very important. We love it. But we learned very quickly that like, yes, discovery, but we have all these other problems actually. So, we have to solve all this other stuff. And we backed our way into Unmixing as like the last problem before you hit the instrument. And it was literally the key, I think, to everything. So we saw with a lot of our customers poorly unmixed data, where it was compensated after the fact to make up for the poor unmixing. And again, Evan took a step back and said, what the heck is unmixing actually doing? What are we trying to calculate here? And we kind of came to the conclusion that probably the right way to do unmixing is cell by cell, which is what our product Ozette Resolve does. And the way to think about it is, every cell in your multicolor sample, it's going to have a slightly different fluorescence spectrum, right? And they're going to combine to give you that multicolor fluorescence spectrum, and it's going to have different autofluorescence properties. But what do we do when we unmix today? We kind of try to pick the best single color representation from all of the single color samples. And then a lot of solution providers will give you some number of like autofluorescence correction channels where you can say this one's going to be for monocytes, this one's going to be for macrophage granulocytes, you're going to get one for something else. And that kind of tries to deal with some of that variation. But we found that the variation that even that you get on the single cell level is enough to cause problems. And that's really the source of a lot of the unmixing artifacts that we see in data. That and spectral overlap between fluors, which the source of the problems with that is more of a mathematical one in the math of unmixing. And that's another thing that we are doing differently. So you sort of combine all of that, slightly different unmixing model, slightly different math, and we're able to do cell by cell unmixing that accounts for the unique autofluorescence properties of every cell and the unique spectral properties of every cell. We're able to look in your single color samples and we're able to find and filter out, those perfectly stained cells whose, properties are representative of the sample because you've done your controls correctly and you're using the same matrix and all that kind of stuff as you're doing in your multicolor sample, of course, right? Otherwise, it's not going to work. And then you're unmixing every cell correctly. And so you're not going to get unmixing artifacts. You're not going to get over unmixing or under unmixing. And if you do, you can typically go back to those single color controls and find that there's a culprit, that you're not actually picking the right cells, the right representative positively stained cells. You've actually got some contamination, and you can use Resolve to filter that out and clean that up so you don't have to rerun your controls and kind of provide that single cell level view of what's going on in your single color controls, which is very powerful to get around problems like carryover, right? As you're, going with your pipette and whatnot. So that's kind of the idea behind Resolve. Save a lot of time, unmix correctly. Again, the investment is in upfront in generating the right controls and creating your, nice clean set of controls. And then you can use that to unmix your data sort of going forward. You don't have to necessarily generate new single color controls with every run, only when your instrument is like maintained or whatever the case may be, or you probably have some SOP that says, I don't know, every month or 2 months, I'm going to regenerate new single color controls. So that's kind of where Resolve fits in. And when it's done correctly, you don't have to compensate.
Well, Ozette Resolve at this point, it only works with like a single instrument, like manufacturer's data, right?
No, actually, we're at the point now we work with the top. It'll work with the top 3. It'll work with Cytec. It'll work with BD. It'll work with Sony.
So then surely you have data on who is the easiest to unmix.
It's all the same. It's all pretty much the same. A lot of it comes down to generating the right controls
in the right or representative matrix when possible. Yeah.
Sounds like you got some fun secret information there, though, that if you already can pick up on carryover from sample to sample, that surely you've identified. You don't have to say what the trends are, but you found some interesting trends.
There's a statistic I throw out that says 90% of flow studies have undetected data quality issues, and I stand by it.
Mm.
How impactful those data quality issues, I think, depends on what that data is used for. But certainly in the pharma setting, it can be extremely impactful. And I think it's one of the reasons that flow has not risen to the utility that I think it has its potential to, as a true, like, first-rate primary endpoint tool. It's still kind of used for secondary exploratory endpoints, but I think it has the power to be a true diagnostic power player.
So you think that there's a definitely potential podcast on how to fake your flow cytometry data with bad practices?
What an interesting concept.
I'm trying to figure out what is the difference between Ozette's approach and what we currently do with our current mixing methods with the platforms that we have. Is it like currently we just basically gate on a cluster of cells, we figure out some like matrix to apply, and it's basically the same value applied to the same cells regardless of, and so you guys found a way to,
that's
like,
what happens today, right?
So you guys found a way to, like, kind of calculate, I'm gonna blow this up, but basically calculate a matrix that's applied to every specific cells. And
yeah, you can think of it as every cell gets its own unmixing matrix that's optimal for that cell.
Okay, that is crazy. Okay.
So roughly you're looking at the raw spectrum of the cell, you're looking at the reference spectrums individually, and then figuring out a min-max combination possibly of what those signals are that overlap to fit, and the remainder is probably autofluorescence.
To some approximation.
For general audience.
Right. Me.
It does really get at the crux of one of the, like, deep pain points in unmixing, which is no two cells are equal. And so when we think about autofluorescence and really teasing apart true signal from artifact or, noise in the system, that becomes really important. How do we account for autofluorescence, and how do we get to true signal? And I think that's one of the biggest differentiators for Ozette Resolve compared to methods that are available today. And yes, every Spectral instrument has their own flavor of an unmixing solution, but at the core, they're very similar, and that, they deal with autofluorescence, I think, very similarly, or, the data aren't perfectly unmixed, and then we have to do things to adjust the data or manipulate the data through compensation, which I don't think anybody really loves to do. And so, yeah. And then
sure. Are you probably the closest then on the market to getting or extracting out a cell by cell, like, unique autofluorescence signature? Because I find
we do that currently. That is exactly what we do.
I mean, again, if I find the whole concept of autofluorescence fascinating, and again, I think there's so much potential there for autofluorescent phenotyping. I mean, would you be able to speak to just, and I think that would speak to the power, potential power of the platform. How much just variability are you seeing in that, or just the potential in itself of autofluorescence? Or one of my notes in here, the ability that which you can take that and apply it to unmixing of fluorophores. Can you take that to the next step? If we gave you reference signatures for NADH, FAD, different flavins, like, are you at the point where you can start unmixing autofluorescence into its base components?
Fascinating. We haven't tried that. But I could see how the approach could be adapted to do that. If you treat those as sort of your pure representation of autofluorescence, and yeah, I think that would be a fascinating experiment, but we haven't tried it.
Can you speak then to just the diversity in which you see from autofluorescence? We did a big autofluorescence workshop this year at Cyto, and we anticipated that there were only 10 other people in the world that cared. We ended up with a few more than that. But it still seems such a point of contention of people being like, it's all just one signal, versus, just tied to how diverse and how powerful is this? I'm right, aren't I? Like that's what I'm asking.
I think you're yeah, I think you're correct. Like we do insist for people using Resolve that if you're you run your single color controls and then you're going to have some unstained control. But then if your samples that you're running in your multicolor are, again, in a different matrix, have different autofluorescence properties because of that, or different fluorescence properties because of that, Resolve provides you with the ability to use a different unstained sample that is more representative of that matrix. And in fact, it's necessary to get the correct unmixing. So it has a very significant impact on the result. It's definitely not just one signal.
And if I'm hearing together overall with the Resolve, because I'm always trying to put these back into context for myself to make sure, what am I not understanding? What am I assuming here? The total package so far is, if I'm building this stuff on my own in R, I've got my one flow AI algorithm, I'm trying to clean stuff up. Then I'm going into my clustering, I'm trying to figure out, all right, what clustering algorithm am I going to use? What scaling am I going to use to get these things correctly? Okay, now what actual, dimensional reduction tool am I going to be using? And it sounds like you guys just went through every single one of these approaches and said, how do I do this better than anything possible? And then through those steps, it's the same thing that we're all stumbling through right now, which is, oh my gosh, the day-to-day variability and just your pipetting from sample to sample is far worse than any unmixing issues or any of these other issues. And you found that quite a good deal about that you can kind of account for. There are still other things that you can't account for, but it was trying to streamline that process a little bit, which as I'm working on training users, I find is an incredibly time-consuming hurdle when they know what they want and roughly what the data should look like. And it's just trying to take that middleman out in a very concise way.
Yeah. I mean, you definitely hit on like all of the important pain points. And as you can imagine, errors that happen way upstream, as you're generating the data, or whether it's in unmixing, or whether it's in, the cleanup that you do, whether it's with flow AI, as you mentioned, or whether it's in your like pre-gating, maybe you have a few hundred samples and you're deciding, I'm just going to drop templates on here. And then, you kind of give it a good look and like, I think it's okay, but there's some small number in there that has a batch effect that you didn't notice for whatever reason. And by the time you get downstream to the end, you've done your clustering and you're starting to like now look for patterns. By the time you find something, right, like it's kind of too late, right? You've done however much work. And so what the platform provides for you is a way to find these things ahead of time. One of the things that it enables is to take in your experimental design into how it runs. So if you have longitudinal data, if you have actual batches of subjects run, you can sort of enter that information and it will take that into account as it's kind of learning about how all of these markers are different and these samples are different. And so when it does the gating, it will adjust for that, right? And it will like kind of look for variability within batches. So it's going to try to look for actual biological variability instead of batch variability, as one example. But at every step, you have the opportunity to like pause and then use the platform to actually get your eyeballs on the data and see, are these gates correct? Is my unmixing correct upstream of that, looking at n/1s and intervene. And so we've spent an incredible amount of effort and engineering resources to build a platform that actually presents you like live single cell data. So all of the visualizations in Ozette are generated live from the single cell data, like as you're sort of querying it and exploring it. We're not like pre-computing, static images. And so you can really use the platform to explore and look at those things that, that are important to you and catch problems, that might be things that about the way a particular marker behaves. And so you have to like look at it at a certain point, you know, and you can bring up, you know, visualizations for 50 samples, you know, basically in an instant. You get kind of everything in one, and we're really good at pattern matching, right? And so you can use that opportunity to look for things that stand out, look for things that aren't quite right. You don't have to resort to like PDF printouts or these like predefined layouts and things like that. You can kind of like really leverage the power of really great visualization and scalability to effectively look at all of your data really, really quickly.
Very nice.
Highlight what Greg mentioned, which is I think we were really intentional about not only advancing the technology for unmixing or analysis, but also I think making sure we had benchmarks and also visibility into quality along all of the tasks over the entirety of the workflow, so that you don't get halfway down the workflow or to the end of the workflow and say, oh no, you know, something's wrong here, and now I have to go back 2 weeks and start from the beginning, maybe possibly by generating new controls, right? And so we have tried to build intuitively in that sense, where you're not getting too far without some quality checks and confidence that you're ready or can move to the next task in the workflow. And then
that's incredible in itself, right? Like most of my arguments, you know, are like you're not ready for this. Like you're not ready to start analyzing, like, you know, the data is poor in this regard or that regard. That's a difficult task that you took on, and a real benefit that you put in there. So I appreciate you bringing that back up.
Yeah, I think for us, you know, there's nothing more important than quality and accuracy of the data. And so, and, you know, I know we touched on automation and training data, and even in this regard, we do view it as, you know, a helpful tool or, you know, an efficiency gain. And the way we think about it is like really turning the user into a reviewer, but you are the ultimate like gatekeeper. You can review and edit, right? And I think that's really important. Like we're definitely trying to streamline, to standardize, to make things reproducible, you know, across all your analyses as a user, but also your team members or a team you're collaborating with. But ultimately, you know, you have the editing privileges. And so, you know, everything is double checked by the expert in the room.
I mean, this is all cloud-based, correct?
Yes.
Correct.
Is that something then I'd love to be able to follow up with you guys at another time, and just if you guys have aggregate data on what those benchmark failings are, I feel like that would just be really interesting as well, just to educate the community that in general, this is something that needs addressing, right? That we're seeing that batch effects are the biggest issue overall, or these things, just again, for community education. Yeah, so many things I want to ask you, but I keep interrupting David, so.
I'm going to switch to the last part where we basically want to discuss just how, like, what are your favorite clients? And if we're a core facility, just how do we interact with Ozette, and how do we put you in touch with our users? So again, Danielle, it's your chance. Just like swoop in with one more question.
Oh gosh, one.
Or more. It's fine.
I mean, obviously, I got to ask. My favorite question is: Can you handle the future cytometry imaging parameters? How does that start fitting into this? Because it's the biggest item I hit with almost all of these right now. The fact that the majority of the parameters, you know, exist on a linear scale, where most of these are between 0 to 1 or 1 to 10, that you're now trying to put into this computational data set, and that, you know, it doesn't scale the same, it doesn't look the same. There's an overwhelming amount of parameters that are telling you the exact same thing and that are biasing your populations of clusters. But it's also the thing that number one right now we need as a discovery tool, because we know it's so powerful. It's just nobody knows what to do with this data right now.
It's not something the platform handles today. I think there's a lot of challenges just with existing spectral cytometry. Certainly, we've pulled in data from, you know, spectral and imaging cytometers. It'll read the imaging data just happily. It'll read those parameters, but, you know, we're not actively using them today. one of the things that um, one of the things that the platform does, sort of when you run Spectral through Spectral data through the Unmixing module, is it'll estimate the appropriate scaling parameters for sort of optimal visualization. So we do have very much the ability to like scale different parameters differently. And so I think given that that's baked in, we could probably extend things in the future to, include imaging parameters. And we've certainly visualized imaging parameters on the platform. But I think, your question stands. It's like, no one's come to us with an imaging data set where we have like some basic, like some outcome that we're looking to hit, right, with that data, like some analytical plan. Here's the things we're trying to learn. And so it's just not a problem that we've tried to tackle at this point. But I'm pretty confident that we have all the tools in place to, we could basically just throw those parameters into the mix as we run the approach. But it's just not something that we've tackled today.
Cool.
So what are your favorite clients when you think about the entire field? Who do you feel you have the best impact with? And then we'll switch to core facilities. Like my core, we don't generate any data at all. So my role dealing with Ozette would basically to link you with other people. So how do you see the roles of core facilities dealing with your group?
My favorite clients are the ones for whom we can solve problems, where we're a solution that makes them more efficient, helps them do better science, solves their problems. That's what I love. And they span everything from individual academics to core facilities to CROs to pharma. We've worked in all those spaces. So I wouldn't say there's a segment, but if we're out there solving a problem for you, it's what I love to see. And if there's feedback on how we can be better, that's an even better customer. Like, I love that. Corrie?
I feel like you said it exactly the way I would say it, which is there's something like deeply satisfying, I think, for our whole team, especially for our team that works with partners on data analysis, to see tangible mitigation or solutions, changes that improve that analysis or analyses downstream for them. And we've had a number of examples of that through Resolve, through Endpoints, through Discovery. And that has spanned academia all the way through different, pharma teams. And so, yeah, I think if we're solving problems or we're making science better in collaboration with our partners, then I think that's the most satisfying for us. For core facilities and how to work with core facilities, one of the things, that is available right now that we're seeing a lot of people interested in core facilities is Ozette Resolve, because it is kind of off the shelf, right? It is something that can be for one user or a team of users. And if you have spectral instrumentation and you have unmixing challenges, essentially the input into that solution is just raw spectral data. And so you can bring your data in, unmix your controls, unmix your multicolor files, and then there is an off-ramp. So if you want to take that into a current analysis solution you have, whatever that might be, in-house, something else off the shelf, there's an easy way to do that. If you want to continue with Ozette into a targeted analysis with defined endpoints, through our endpoints analysis, or something more broad like Discovery, then that's possible also. So we try to make it really easy and kind of meet folks where they are. But I think where we are seeing a lot of traction with core facilities, especially those that use spectral instrumentation, is on the Resolve platform, or on Resolve on our platform.
I didn't check, but you guys have a subscription model?
We do, essentially, yeah. It's based on volume, and we know that, depending on where you are in your project or how big your team is, there can be a variation in volume. And so it is volume based in that subscription or yearly subscription. And so we do try to take into account like where folks are in their work and what is appropriate for that work. Again, you know, to the accessibility piece, like we want everyone to be able to use it, but we also want to make it available to as many people as we possibly can.
Obviously, we talked about flow stometry because we're nerds about flow, but like you cover a wider range of technologies as well. So that should be said at some point. Dan, any questions? Like I think we're reaching the last few minutes together. Any last things you'd like to cover?
Oh man, it's just there's just so much stuff moving to flow cytometry, and it's all going to come back to this: is how do we even start to analyze the data? I know that's the big struggle right now for shared resource facilities. When I talk to other cores, is we all know that we need essentially at this point a dedicated staff to data analysis. And I feel like everybody's trying to come up with a lot of patchwork solutions where, you know, a lot of FASs are working overtime to help people because we all want to see the science being driven forward. And I'm just excited that there are more tools coming out to try to make this something that is easier, that is more, you know, accessible, and wherever that's going to take. You know, I'm excited for what Lorraine and Doug was talking about with his ludicrous flow cytometry, that there are things we already know we can do. We don't even know how to start analyzing the data on that. There's path forwards on that. That's exciting to me. So thank you for putting this on my radar. I look forward to just chatting more and learning more about what's possible.
Absolutely.
Awesome.
All right. Where do we find you guys? You have your website? You have your...
We have our website. We do. That's a great place. Our team's going to be at IO360 next week, in fact. And then we will also be at Cyto, including probably myself and Greg. Yeah.
Yeah.
So maybe we'll have an
Booth.
opportunity to see each other in person.
All right. Looking forward to it. Greg, Corrie, thank you so much for your time. It's been great. And we'll talk to you guys later. For viewers, leave comments, do the thing with the clicking. And all right, thank you much.
Thanks, David. Thanks, Daniel.